Okay, well, it's a pleasure to be able to give a talk again for the brain cancer course that we hold every year. Sorry, I have to pre-record this because of another place I have to go during the day of the course. But my name is Charles Cobbs. I am a neurosurgeon here at Swedish Neuroscience Institute, and been fortunate enough to be able to run a translational research lab here. And I would like to discuss a model we have in tissue culture that I believe will lend substantial support for the concept of a type of immunotherapy clinical trial that we're trying to develop at Swedish. First slide is to just state that I have conflict of interest with HDT Bio, which is a local biotech company who I'm working with on this project, and I'm a scientific advisor for the company. The essential issue that we're discussing today in this talk is the fact that immunologically, some tumors are cold and some are hot. Hot tumors like melanoma and some lung cancers have a lot of mutations, and that can be leveraged by using checkpoint inhibitor drugs to amplify the attack mode of cytotoxic T cells just non-discriminately. And when you do that, you can attack these tumors because they have so many neoantigens. Glioblastomas and most other solid tumors fall into the category of cold tumors. And glioblastomas in particular are extremely cold tumors. And what I mean by that is not only are they devoid of cytotoxic T cells, but they have molecules that are expressed that promote the immunosuppressive phenotype of the tumor microenvironment. TGF beta in particular is one that we see a lot. And T cells that are in these glioblastomas often have high levels of checkpoints on them, like CTLA4 and PD1 ligand. And so one of the critical issues with glioblastomas is how are we going to get an immunotherapy to work when the tumor is essentially a force field that prevents infiltration of cytotoxic T cells? And so we know that checkpoint inhibitor drugs have not really shown great efficacy for glioblastoma because even when you give them, the T cells don't really penetrate into the tumor. And so part of that problem is that the microenvironment is full of macrophages and other cells that are in the so-called M2 phenotype, which means that they are actively promoting anti-inflammatory and anti-tumor immunosuppressive molecules. I like to go back to this slide. And so what I'm going to do is kind of take you through some concepts, general concepts of immunology and cancer immunotherapy. For years, we've known that most tumors that develop from epithelial cells, and glioblastomas are neuroepithelial, are thought to arise from either some type of inflammatory process or, in many cases, an infectious process. And in most events, when you have an inflammation of an epithelial layer, there are infiltrating macrophages and dendritic cells and monocytes, which are innate immune cells. But then you go back to the normal epithelium. However, in cancers, often you have microbes or chronic inflammation, and the infiltrating cells essentially do their job, and then you have a dampening of the subsequent immune system such that over time, if there's chronic inflammation or chronic expression of oncogenic elements from something like HPV, for instance, in cervical cancer, then there becomes a point where the microbes induce enough mutations and the tumor microenvironment immune system gets suppressed to the point where you make a transition from chronic inflammation to tumor, and the immune system is not doing its job. And just to go back to the basics of immunology, but it's very critical for the concepts of this talk. There are two arms of the immune system, the innate immune system and the adaptive immune system. Consider innate immune system function to be that function which has been pre-programmed genetically into you from millions of years of evolution. So that if your cells are exposed to certain molecular things like double-stranded RNA, which is specific for viral infections, or certain molecules that are seen in pathogens like bacteria, like lipopolysaccharide, then they have ingrained, million-year-old mechanisms to activate huge amounts of certain cytokines that will bring in immediately macrophages and dendritic cells and NK cells. That occurs in the first few minutes to hours of this exposure. And then these cells will then pick up little pieces of various parts of the cells that are infected or destroyed, go back to the central processing unit of the immune system, which is the lymph node or the spleen, and they will direct these so-called adaptive cells, B cells and T cells, tell them to expand and focus in on specific molecules, and that's the adaptive response. And it's thought that in cancer, there is probably an initiating event where you have a massive induction of innate immunity, and this is associated with these ingrained molecular pathways, and two of the sort of hallmark pathways are called RIG-I and toll-like receptor pathways. These pathways go up immediately upon exposure to a pathogen or some type of cellular damage. That leads to secondary infiltration within days to a couple of weeks of the adaptive immune system, where you may get massive expansion of specific clones of T cells that are specific to these molecules. These guys come in, and it's thought that during the evolution of cancer, for instance, if a pathogen is involved early on, you get an elimination of that pathogen. However, back to the HPV and cervical cancer analogy, if that pathogen is not completely eradicated or there's multiple rounds of infection over time, then that pathogen can alter the genetics of the cell and maybe make those cells become resistant to the effects of the T cells, and they can actually reprogram those T cells to go from cytotoxic T cells that are active to cytotoxic T cells that are, quote, exhausted T cells that have immunosuppressive functions, and they can even change the function of the infiltrating macrophages, et cetera, to make the environment for the tumor immunosuppressive. And that's called the equilibrium state, which leads to escape, where the immune system in that tumor is actually promoting immunosuppression, and it's allowing these tumor cells to outgrow, and that's when cancer rises. In the last several years, there's been an increased interest in the possibility of leveraging sort of the innate process of your immune function for various tumors, including glioblastoma, and this is from a review article. The concept is, if we don't have specific antigens in glioblastomas that we can target with CAR T cells, whatever, et cetera, can we leverage that process of activation of the innate immune system to reboot the immune system to get it back to functioning? And so, as I mentioned, there are innate factors in the cells in a tumor or anywhere that are specific and responsive to pathogens and to damage. These are so-called pathogen and damage-associated molecular patterns that I briefly mentioned. Would it be possible to take a glioblastoma, which is a cold tumor, and insert something or do something to the tumor that would dramatically induce these pathogen and damage-associated molecular patterns and essentially reboot the immune system? And, if the tumor is in this state, drive it back so that you have a massive innate immune response, call in a whole new squad of infiltrating T cells and immune cells, which would look at the tumor for the first time in a long time as a abnormal environment. And there is evidence in that setting that even if you don't have specific molecules you're targeting, if you can induce that response to the tumor, and especially if you can do it in the setting of a pathogen, then T cells may come and attack the pathogen. And there is something that is a very interesting phenomenon called epitope spreading, that in that environment, all of a sudden, these infiltrating cells may also say, hey, there's a tumor here, and they may reboot the original anti-tumor response, and so you can get clearing of the tumor. So just to briefly discuss a couple of mechanisms that are used to activate innate immunity. There's a molecule that's been around for a long time called poly-IC, and it's basically a synthetic molecule that looks like double-stranded RNA. And as I mentioned, when you have double-stranded RNA, it can activate both the RIG-I pathway and the toll-like receptor pathways. And these are two evolutionarily conserved pathways in all mammals that dramatically activate interferon activation, which sort of reboots and re-establishes the innate immune activation of the tumor, and that would lead to increased inflammatory cytokines, increased dendritic cell infiltration, and reshifting of that immune suppressive microenvironment to a more anti-tumor microenvironment. And people have been studying viruses that are associated with glioblastoma. One that my lab has been involved in is cytomegalovirus, and they've shown that if you have a person who's been exposed to cytomegalovirus or an animal, then you can actually, by rebooting this innate immune system in the setting of adding viral peptides that would call up a established immune response, you can dramatically increase the effects of this for the effect of the activation of the innate immune system, essentially trying to artificially produce innate immune activation and the setting of a viral infection to really bolster that innate immune function. So it's not just theoretical. There have been a few papers over the last few years that have investigated this concept called peptide alarm therapy. One of the key investigators is at the University of Minnesota, Dr. David Massafust. But this paper came out a couple of years ago that was really striking to me by Dr. Schiller at the NCI, who's a leading cancer immunotherapy researcher at the National Cancer Institute. They decided to study the activation of the innate immune system in the setting of recall for cytomegalovirus using solid tumors of different types in sub-Q model of mice. And essentially, they gave mice cytomegalovirus infection at birth, waited 6 months, and they implanted tumors into the mice. And they gave various combinations of either nothing, saline, and all the mice died at essentially day 30, or they gave poly-IC, which is the molecule I mentioned a minute ago that makes the cells think that there is a double-stranded RNA, and it induces gamma interferon in the innate immune response. When they gave that alone, it did increase the survival of the animals. However, if they gave the poly-IC plus specific peptides that the T cells for that mouse had been trained on for the cytomegalovirus infection, kind of a lot going on, but basically they added peptides to the tumor microenvironment, and it allowed the T cells that had already been established and were memory T cells to see this pathogen and activate those T cells in the setting of activation of the innate immune system. When that happened in combination, there was a profound anti-tumor response that was not just against the pathogen, but it was also able to eradicate the tumors. None of the mice died in any of these situations when they received activation of innate immunity by poly-IC plus recall of a prior infection using peptides against CMV. And it's worth noting that in humans, for whatever reason, about 10% of a lot of people, even up to 30, 40% of some people's complete T cell memory pool is specifically dedicated to cytomegalovirus. And that virus, is common in most adults. About 80% of adults have been exposed to CMV. So my lab, which has already been studying CMV and glioblastoma, we had a slice culture up and going that we were using to do other types of studies. And basically, in this situation, we take out a tumor and we can slice it in 350 micron sections, and these can survive for several days in tissue culture media. And we can study them. And this is just a H&E and a CD8 cytotoxic T cell stain. where you can see there are CD8 cells in the blood vessel here, but hardly any cells exist in the tumor. And that's just classic for glioblastoma, where there is essentially no active immune function going on in the tumor. And that's so-called cold tumor illustration. About this time, and about the time I had read that paper from the lab at the NCI about activation of innate immunity in the setting of CMV, I was lucky enough to be introduced to some colleagues that work at a local biotech company that's one of the leading makers of RNA vaccine technology. They have a proprietary molecule with a lipid core surrounded by RNA that is made to not only go into a cell specifically, but also to amplify the signal on the RNA. And one of these molecules is a molecule that activates the the exact same thing that you see with poly-IC. So it is a profound activator of toll-like receptor 3, giving that signal that there's a viral infection to the innate immune system. So essentially, if you inject this this molecule, it goes into cells, and then they start expressing high levels of the molecule of interest. And we tested it in glioblastomas by first using a molecule that they have that expresses GFP, EGFP, which is a fluorescent marker that turns green. So we would take out a glioblastoma, and on the chunk of tumor would be injected with this molecule and sit overnight, or else cut into slices, and we would put the molecule in the media, and we could see a day or so later that there was diffuse and high levels of expression of the GFP green fluorescent protein throughout the glioblastoma from a patient. So we had the concept that what if I took out a tumor and we wanted to test the ability to activate the innate immune system and do that in the setting of recall of CMV peptides for a patient. And of course, the patient needs to have been exposed to CMV, which is not hard because, as I mentioned, 80% of adults are positive. So we have done several experiments, and I'll illustrate one of the results that we had based on this study. And I think that this is quite exciting and could lead to a rational basis for clinical trial. So essentially what occurs is we take out a glioblastoma, we put it in a little thing that looks like a sausage slicer and slice out 350 micron pieces of the tumor, and these go into tissue culture. And then in that setting, we can either treat it with just nothing or with the HDT-401, which is a lipid RNA molecule that gets into the cell and produces high levels of essentially a signal for toll-like receptor 3 to activate innate immune function. We can do that alone, or we can do that with adding in peptides of cytomegalovirus. And this patient that we did it with was CMV seropositive, so they had a prior immune system response to CMV. And then we also did it after doing that, plus adding some of the patient's PBMCs, the white blood cells into the media to see if the tumor cells could express certain things that would be amplified by actual immune cells being in the setting of the tumor. And this is an illustration of some of the cytokines that were studied. This is just one case, but I think it's exciting to show some of the things going on. Some of the critical cytokines that are involved in taking a cold tumor and turning it into a hot tumor are inflammatory cytokines like IL-6, tumor necrosis factor, interferon gamma, IL-12, and GM-CSF. This is actually a cytokine that is immunosuppressive. So if this goes up, it will make the tumor more immunosuppressive. And if it goes down, it will make the tumor less immunosuppressive. So what did we find? The very interesting thing is that at the baseline, without any treatment, these inflammatory cytokines, IL-12, gamma interferon, at day one is in black and day 3 is in gray. At baseline, oops, let's go back. At baseline, there's hardly any expression of inflammatory cytokines. If we were to add the HDT401 activation of toll-like receptor 3, you do get induction of IL-6. You get pretty significant induction of tumor necrosis factor. You get some interferon gamma production, but not so much from IL-12 or GM-CSF. However, if we give HDT401 plus the viral peptide, we get increased expression of some of these inflammatory cytokines. But what is really exciting and really striking is if we give all three HDT401, CMV peptide, and PBMCs, you can see that at day 1 and day 3, you start getting a much higher level of expression of these cytokines. For instance, with all three of those constituents, you get a 1,500 fold increase in IL-6. After 1 day, you get 400-fold increase of TNF and gamma interferon, essentially several hundred-fold increase in gamma interferon by day 1. So this is consistent with activation of innate immunity in the setting of viral infection and activation of innate signaling pathway to like receptor 3. And I'd like to kind of hone in again a little bit on just gamma interferon. As I mentioned, gamma interferon is one of the most critical activators of innate immune function, and it has the ability to really alter the tumor microenvironment to make it much more hospitable to inflammation, infiltration of dendritic cells and macrophages, and alteration of those cells from an immunosuppressive to immune activating phenotype. So just this dramatic increase gamma interferon by the addition of this RNA molecule plus CMV peptides in the patient's own white blood cells is quite striking. And just to show, this is a schematic of what gamma interferon can do to a tumor microenvironment. Macrophages, which are often seen in in the glioblastoma microenvironment, which are immunosuppressive, can be reprogrammed to become inflammatory. And this also can induce upregulation and maturation of dendritic cells, infiltration of dendritic cells to really reboot the immune system. And all of that can induce infiltration of T cells to activate more of an antiviral response. And in that setting, we believe it would lead to also this off-target anti-tumor epitope spreading response. So based on these studies and some animal work we're doing now, we are working with HDT Bio to try to come up with a phase 0, phase 1 protocol for clinical trials where we would potentially do a brain tumor biopsy, for instance, in a recurrent glioblastoma setting, and inject in doses, increasing doses, the HDT401 molecule, and then resect the tumor and see if we can get infiltration of cytotoxic T cells and increased activation of cytokines. Simultaneously, we would do a dose escalation of CMV peptides in seropositive patients, and then consider combining these at some point to see if we can essentially recreate in a patient what we have been able to recreate in a dish and in animal models, which would be a alteration of the cold microenvironment to a hot microenvironment, and hopefully anti-tumor response akin to that seen by the NCI group that is doing these similar studies. So couldn't have been done without a huge amount of great work from the folks in my lab, Dr. Hoti, Yoon, Dr. Lee, and several of my colleagues at HDT Bio and other members who've helped out. And this is supported so far by primarily the Susan McGregor Immunotherapy grant that we have, and a recent grant that we just got, as well as the Paul Allen Research Award. So thank you very much.